Hello, everyone, and welcome to today's webinar, Applying Whole Genome Sequencing to Define and Predict Antimicrobial Resistance. I am Marie Stone of Laboratories, and I'll be your moderator for today's event. Today's educational web seminar is presented by Laboratories and brought to you by OpGen. To learn more, visit opgen.com. We encourage you to participate today by submitting any questions you may have during the presentation. To do so, simply type them into the Ask a Question box and click Send. We'll answer as many questions as we have time for at the end of the presentation. You may also submit any technical issues here as well if you have trouble seeing or hearing the presentation. This webinar is educational and thus offers free continuing education credits. Please click on the Continuing Education window at the bottom of your screen to obtain your credits. I'd like to now welcome our speaker, Trish Simner, PhD, Associate Professor of Pathology, Johns Hopkins University School of Medicine, and Director of the Medical Bacteriology and Infectious Disease Sequencing Laboratories, Johns Hopkins Hospital. Dr. Simner, you may now begin your presentation.
Thanks so much for the kind introduction. So this afternoon, we're going to be discussing using whole genome sequencing for prediction of antimicrobial resistance. Here are my disclosures. I do have a pertinent disclosure for this presentation. I am a consultant for OpGen, for which I will be presenting research with Ares Genetics today, which is a subsidiary of OpGen. So the objectives for today are to describe how whole genome sequencing can be used to define mechanisms of antimicrobial resistance, understand the advantages and disadvantages of using whole genome sequencing to predict antimicrobial resistance compared to phenotypic antimicrobial susceptibility testing, and discuss the status of applying whole genome sequencing to predict phenotypic AST for clinical care. So the threat of antimicrobial resistance is one of the biggest global public health threats that we're currently faced with. It is recognized by many international bodies as a priority to address, as it's a leading cause of death worldwide, where its highest burden is in resource-limited settings. Despite understanding that it's a leading cause of death, the precise magnitude is not well understood. The most recent estimates on mortality associated with antimicrobial resistance was just published in Lancet in January of this year, where they estimated in 2019 that there were 4.95 million deaths associated with antimicrobial resistance, and within them there was 1.27 million deaths attributed specifically to bacterial antimicrobial resistant infections. And so global collective action is required to address this threat. And two of the initiatives that have been described to address the threat of antimicrobial resistance include improving global surveillance for antimicrobial resistance and promoting new rapid diagnostics to reduce unnecessary use of antimicrobials, both of which can be addressed through whole genome sequencing applications, which I will discuss throughout my presentation. So the standard method for clinical laboratories worldwide to detect antimicrobial resistance is through the standard identification and antimicrobial susceptibility testing methods. On average, these methods take between 2 to 3 days, where we collect a specimen in the lab, we'll plate it to appropriate media. From there, we will isolate the organism on the following day after overnight incubation. We will perform MALDI-TOF mass spectrometry for identification of that organism. And if we have pure and sufficient growth, we'll set up our antimicrobial susceptibility testing panels. The following day, our standard AST panel results will be available. And if we encounter a multi-drug resistant organism, we may need to set up additional antimicrobials or send out to a send-out lab if testing modalities are not available within that lab, further delaying those antimicrobial susceptibility testing results. Furthermore, if you're dealing with a messy culture, this might delay the turnaround time, or dealing with a more fastidious organism that might have a slower growth rate, this may result in longer turnaround time. Luckily, throughout the last decade, and we've seen the introduction of many new tools that can help us more rapidly detect antimicrobial resistance in the lab. These can include simplex or multiplex PCR panels that can be performed directly from the clinical specimen, or from cultured isolate recovery of that cultured isolate on solid media. Alternatively, there are rapid phenotypic methods that can be applied, whether that be chromogenic agars or rapid phenotypic tests like the PBP2A test that can be performed from culture growth of Staph aureus to predict whether the organism is methicillin resistant or susceptible based off of the PBP2A assay. Furthermore, we started to see the introduction of rapid phenotypic comprehensive AST panels that can be performed from positive blood cultures that allow us to more rapidly detect the phenotype using novel methods for detection like digital microscopy or microbial weighing to more rapidly discern that phenotype than visible growth. In addition to these methods, we're starting to see the introduction of whole genome sequencing to predict phenotypic AST. The advantages of whole genome sequencing is that we're starting to see the methods become more rapid and can be used in a clinically relevant timeline, as well as it detects this comprehensive phenotypic AST. It has the potential to detect a comprehensive phenotype. So what is whole genome sequencing? Well, it's an application of next generation sequencing where you're sequencing the genome of a pathogen of interest. And you'll sequence and sometimes just taking the reads, or you'll assemble it. And from that, those reads or that assembly, you'll then detect the antimicrobial resistance genes associated with that organism. And all the antimicrobial resistance genes or single nucleotide variants associated with resistance is referred to as the resistome of that organism. You can then use that resistome or the entire genome of your organism to define mechanisms of resistance or to predict your phenotypic antimicrobial susceptibility and resistance phenotype. And so that's what we'll be discussing today. So we're going to go over these two applications of whole genome sequencing for detection of antimicrobial resistance. So the first item we're going to address is using whole genome sequencing to decipher mechanisms of antimicrobial resistance. And to do this, we're going to do it through a case presentation. So this is a 35-year-old woman living in the United Arab Emirates who was on a flight to the United States to seek medical care for a relapse of her leukemia. During the flight, she became febrile, tachycardic, and experienced lower abdominal pain. In the emergency department, a blood culture was obtained and she was promptly started on cefepime. However, her hospital course was complicated as a urine culture on day one and imaging revealed multiple intra-abdominal abscesses that were drained, and both the urine and intra-abdominal abscess grew the following E. coli with this multi-drug resistant phenotype, where it only remains susceptible to amikacin and gentamicin. Based off of this profile and the fact that it was resistant to both ertapenem and meropenem, we would set up the CARBA-5 assay to try to discern what is the mechanism mediating carbapenem resistance in this isolate, and it was found to be an NDM-producing E. coli. When we see these complex or these multi-drug resistant phenotypes in the lab, we then start to release additional agents or cascade report additional agents on our panels, as well as set up additional antimicrobial susceptibility testing to try to guide therapy. And in this case, this isolate was also resistant to the novel β-lactam, β-lactamase inhibitors like ceftazidime, avibactam, meropenem, faverbactam, and imipenem, relbactam, all consistent with this being an NDM-producing E. coli. It did, however, retain susceptibility to cefidorocol as well as minocycline and tigecycline. And so the patient was transitioned to cefidorocol therapy on day 3 once these AST results were available to the clinical team, and chemotherapy was initiated on the same day. So what is cefidorocol? You might be asking. So cefidorocol is a novel siderophore conjugated cephalosporin that uses this Trojan horse approach to gain entry into the bacteria. So the siderophore component will bind free iron, and once that iron is bound to the agent, it can then use iron transporters for entry into the cell. Once it's across the outer membrane, it disassociates from the iron molecule and binds its target, PBP3, disrupting cell wall synthesis. The unique thing about cefidorocol is that it has broad activity against multi-drug resistant gram-negative organisms, including metallo-beta-lactamases like our NDM-producing E. coli. So the patient unfortunately developed new fevers on day 17, and tigecycline was added to the cefidorocol at that point. And on day 22, a urine culture revealed 2 morphologies of E. coli after 19 days of cefidorocol exposure. And the first morphology demonstrated a slightly elevated MIC to cefidorocol as well as minocycline. However, the second morphology demonstrated frank resistance to cefidorocol as well as minocycline and tigecycline. So while on cefidorocol and tigecycline therapy, the patient we further recovered the same second morphology listed here from the blood culture isolate of this patient. So the question that came up was, what is the mechanism mediating resistance? So we've seen the development of resistance to cefidorocol well on therapy in this patient. And so we used whole genome sequencing using both long read and short read sequencing approaches. So we used the long read approach to allow us to get closed whole genome and plasmid assemblies. And we used the short read Illumina sequencing to help us kind of proofread the longer reads generated from nanopore sequencing because they have a higher error rate than does Illumina. So it allows the Illumina reads allows us to kind of proofread our assemblies. And so we generated these hybrid assemblies for the 5 isolates and demonstrated that and performed whole genome sequencing and demonstrated that all isolates belong to sequence type 167. And all isolates also contain this multi-replicon INC-F plasmid that at a minimum harbored 1 copy of the NDM5 gene. However, we did see increasing mobilization of the element harboring NDM5 within that plasmid from up to 5 copies in isolate number 4. So that was our second morphology from the second urine culture, and then up to 10 copies in the last isolate that we recovered from the patient's blood. And the number of NDM copies correlated with the increasing cefidorocol MICs that we were observing clinically. So we have this association with increased copy number and increasing cefidorocol MICs. So to prove that that the increased copy number was associated with cefidorocol MICs, we did the increasing cefidorocol MICs. We did additional studies, including expression studies, where we demonstrated increased expression up to 2- to 4-fold expression in the isolates with 5 and 10 copies of the NDM5 gene. Furthermore, we demonstrated it phenotypically that we can inhibit the increased resistance by inhibiting the NDM gene. And we did this by using a divalent chelator, EDTA. And metallo-beta-lactamases like NDM require zinc at their active site for activity. So if you chelate zinc and divalent cations, then the NDM will no longer have activity. And so for each isolate, we set down a cefidorocol disc with and without EDTA. And you can see for the first 3 isolates where we have only a single copy of the gene, we don't see any or very little effect of adding EDTA to that disc. Whereas in isolate 4 and 5, we start to see expansion and opening of that pseudochol zone diameter consistent with when we inhibit the NDM, we see restored activity of pseudochol, and further supporting this association that this increased copy number NDM5 led to increased expression and leading to pseudochol resistance. However, the larger question still remains, is this increased copy number the only thing driving this increased resistance that we were seeing clinically? So we've studied mechanisms of pseudochol resistance in my lab through various different studies. And on this slide, you'll see pseudochol depicted here in these light blue circles. And when it chelates to the iron on the left-hand side of the slide, it will then allow for entry through the iron transporter of pseudochol into the periplasmic space, where it will disassociate from the iron molecule and then be able to bind the green PBP3 depicted here and disrupt cell wall synthesis. However, what we found through various studies, either looking at baseline pseudochol resistance, so this is resistance before the agent was even available clinically among multi-drug resistant gram-negatives, as well as additional studies, such as the one I'm presenting here with the development during therapy, and other case reports, is that we found that there are various different mechanisms that can mediate pseudochol resistance, including mutations within iron transporter genes, the increased expression or modification of beta-lactamases. So this case is an example of increasing the expression of that beta-lactamase to then provide resistance or elevated MICs to pseudochol. We've also seen modification of AMP-C among inducible AMP-C beta-lactamases, leading to resistance with both Enterobacter cloacae and Pseudomonas aeruginosa. Additionally, there's been descriptions of mutations in the component of the intermembrane protein complex providing energy to TOMB-dependent transporters like iron transporters, which can lead to elevated pseudochol resistance, or in the two-component transcriptional regulators that have been reported to affect the expression of iron transporters. So in this case, we wanted to look further to see if there was any other potential mechanisms that we had previously seen contributing to the resistance that we've seen develop in this patient case. And so we did variant mapping against isolate 1. So isolate 1 was our first index isolate with a single copy of NDM5, where our pseudochol MIC was still sitting at 2. And then we took all consecutive isolates that we recovered from the patient after that and compared it to the index isolate to see if we can find any consistent mutations or additional mutations leading to resistance in these isolates. And there was no consistent mutations. We did see a mutation in MZ consistent with what we had seen in the past, but it wasn't present in isolate 4, where we were already seeing frank resistance at an MIC of 32. So we concluded that these additional mutations likely contributed a minor role to the resistance that we were seeing in the development of resistance that we observed in this case. However, it shows you how you can use whole genome sequencing, especially in these cases in the progressive development of resistance, to try to define additional single nucleotide variants leading to resistance. So ultimately, in this case, we ended up using the combination of ceftazidime, avibactam, and aztreonam for therapy for this patient, where metallo-beta-lactamases do not hydrolyze aztreonam. However, in this case, there was additional beta-lactamases being produced that was resulting in aztreonam resistance. So the avibactam would inhibit those beta-lactamases, restoring the activity of aztreonam, and we were able to show in vitro, either by a synergy method and by a MIC-based method, that we predicted that this combination would be susceptible in this case. And that was what we ended up treating this patient with successfully, and the patient is doing well to date. So this is a case where we were able to demonstrate how we can use whole genome sequencing to define mechanisms of resistance. And so how can we relay that information into predicting the phenotypic AST of applying whole genome sequencing to predict phenotypic AST results? So what are the pros and cons of using a whole genome sequencing approach to predict AST? Well, let's start out with our conventional phenotypic AST methods. We know this is a highly standardized growth-based method that determines what concentration of a drug inhibits the growth of a bug, and it's interpreted using clinical breakpoints that have been established for each drug-bug combination using MIC distributions, pharmacokinetic and pharmacodynamic data, as well as clinical outcomes data. The pros to using our conventional phenotypic methods is that they're highly standardized. Our physicians are experienced and confident and reliant on those results. These methods are relatively cheap, and the great majority of laboratories, especially CAP-accredited laboratories within the US and internationally, greater than 90% use automated methods for determining a phenotypic AST profile. The cons to this are that they are slow. As we shown as I shown initially, that it can take 2, if not days, if not longer, for the physicians to get that AST profile to try to help tailor therapy. And furthermore, there's little to no AMR mechanism information that is gained from the phenotype. Whereas with whole genome sequencing, to predict the phenotypic AST, we're using the resistome, or the entire genome of an organism, to predict antimicrobial susceptibility and resistance using a molecular method, and in this case, we're using next-generation sequencing. The pros to this approach is that it has the potential to be more rapid. There are some sequencing methods like nanopore sequencing where you can analyze your data in real time and can allow for a more appropriate or more clinically relevant turnaround time. It also has the ability to identify antimicrobial resistance, and we can interpret those results in the context of the phenotypic AST as susceptible or resistant, so that the clinicians are used to the results that we might be generating using this novel method. And last and most important is that the information that we gain from whole genome sequencing not only can be used to predict phenotypic AST, but it can be used for many other purposes, like strain typing for outbreak investigations, looking at virulence factors for prediction of clinical associations, etc. So there are many different applications to the data being generated that we can use clinically the whole genome sequencing data for, not just for predicting phenotypic AST. The cons to this, though, is that it's not as standardized. There are some complex interpretations, and results may vary based on analysis pipeline and or databases used. It's expensive, especially if you're comparing it to the phenotypic AST methods. These methods are a lot more complex than our methods that we're using currently. And right now, there's really no outcome studies demonstrating the clinical utility of whole genome sequencing AST to predict the phenotype. But luckily, what we've seen in the literature to date is that whole genome sequencing to predict AST has pretty good accuracy. And here's just a summary of some of the studies that were performed. And most of these initial studies were performed with the slower but more accurate Illumina sequencing. So Illumina has shorter, accurate, high accuracy reads, but the sequencing run needs to has a certain sequencing time where the sequencing run needs to be complete before you can analyze those results. And in general, in these initial studies, they were running from 24 to 48 hours before it can even be analyzed. And that does not take into consideration the upfront wet lab work. Most of these studies focus on one particular pathogen with mostly manual curation for predictions. And the studies varied by the methods, the sample size, the antimicrobial agents evaluated. However, what these studies do show us is that there is a potential to apply whole genome sequencing to predict AST, because the great majority of the studies demonstrate greater than 90% sensitivity and greater than 95% specificity. However, the process for whole genome sequencing to predict AST is quite long and complex, as I mentioned, where you might take a pure culture isolate from a solid media plate. Alternatively, you might want to do some sort of liquid culture or inoculum-based approach, like we're starting to see from positive blood cultures, like the sepsi-typer approach, etc. You could take that biomass, pure biomass of your organism of interest. You have to prepare your sequencing library, load your flow cell, start the sequencing run, and start to collect your data. Then you're going to detect your AMR genes and then predict your susceptibility profile. So the problem with this from a clinical aspect is that the methods are complex and the turnaround time can be too long depending on the methods applied. The prediction models are still being developed, and as I mentioned previously, they're often developed for only a particular one organism of focus and not a broad spectrum of organisms. And the third is that we need simple and automated tools to generate these predictions because we don't have the time in the lab to do these manual curations of which some of these research studies are applying. So how do we address these issues? Well, we can address the methods by finding methods to more rapidly set up sequencing, but as well as interpret our sequencing results. And so what's available in the literature about interpreting or applying whole genome sequencing to impact patient care or therapeutic decisions? So I'd like to reference this one study by Shelburne and others in CID. It's from 2017, but it really was the first feasibility study where they used whole genome sequencing to predict empiric therapy for neutropenic fever patients. And so they used whole genome sequencing of 90 gram-negative bacilli, including Enterobacterales as well as Pseudomonas, to predict the phenotype for piperacillin and tazobactam, the carbapenems, cefepime, and ceftazidime. And they took these isolates from bloodstream infections of neutropenic cancer patients. They sequenced the isolates on the MiSeq, and they did manual curation to determine the presence or absence of acquired antimicrobial resistance genes, as well as detections of mutations that can lead to resistance to these beta-lactams. And overall, they found a sensitivity of 87% and specificity of 98%. And so this study really demonstrated the feasibility of using whole genome sequencing to guide antibiotic treatment decisions for patients with life-threatening infections for an array of medically important gram-negative pathogens. So what we wanted to do in our lab was address the time, the turnaround time. So in that study, they were using Illumina. It was a retrospective study. They did manual curation. So retrospectively, they were saying, could they apply this data? But technically, it still was not within a time frame that would be clinically relevant. However, we know with nanopore sequencing, the long read sequencing, the advantage of long reads I highlighted earlier from a whole genome sequencing perspective is that you can get enclosed genomes and plasmids, but also it allows us for real-time sequencing analysis. So as we start the sequencing run, we could already start to analyze the data being produced, unlike the Illumina, which requires you for your sequencing run to be complete. And so this has a benefit of being able to detect antimicrobial resistance genes quite rapidly. And we'd previously shown in another study that within 2.3 minutes of starting a sequencing run, we could detect KBC in a KBC producer. And so in this study, we wanted to see if we can use whole genome sequencing, or this real-time whole genome sequencing approach using nanopore to optimize antimicrobial therapy for multi-drug resistant gram-negative infections, specifically for carbapenem-resistant organisms. And in this study, we focused on Klebsiella pneumoniae and included 28 carbapenem-resistant Klebsiella pneumoniae, 19 of which were carbapenemase producers, and 9 of which were non-carbapenemase producers, including 12 carbapenem susceptible isolates. And so what we did is we sequenced these isolates on the MinION sequencer and took two different approaches to analysis. And the reason why we did this is one of the major pitfalls to nanopore sequencing is that it has a higher error rate. And that error rate becomes relevant when you're trying to predict single nucleotide polymorphisms leading to resistance. So we took these two separate analysis approaches from the data generated from nanopore to see how accurate our predictions could be for phenotypic AST. The first approach is what we called the real-time analysis approach, where we just took the sequencing reads coming off the nanopore and mapped them against a resistance database, which is the CARD database using the ARMA tools that are available with nanopore sequencing. And with our methods, we can, from isolate to results, we can get results within 8-hour turnaround time. So making it relevant to clinical care and more rapid to phenotypic AST, where in general you'll still need that overnight growth or 24 hours before you get those results. We also took an approach to an assembly-based approach where we tried to assemble the genome using consensus to try to correct for some of those errors that we know are more error-prone with nanopore, but also applied some bioinformatic tools like nanopolish and additional tools to try to correct those errors to get a better prediction, especially for those single nucleotide polymorphisms. However, with that initial bioinformatics in the background, it added 14 hours to our turnaround time. However, overall, we did show that these approaches did pretty well compared to our reference broth microdilution AST results. Not surprisingly, the real-time approach only gave us 75% overall agreement, and you'll see that the agents that are resistance is mediated by single nucleotide polymorphisms like the fluoroquinolones, ciprofloxacin or levofloxacin here only had 30% agreement, and that's because that was the real-time approach where we weren't trying to correct for some of the nanopore errors. When we took the assembly approach, we've seen a lot better agreement at 91% and started to see better agreement for even predicting some of those single nucleotide variants leading to resistance. And so showing us that even though there's a higher error rate with nanopore, you can use bioinformatics tools to kind of overcome that error rate to allow us to apply this methodology to make a difference clinically, where we can have these results in a clinically applicable time. So here's just showing what the standard turnaround time would be from a blood culture with our phenotypic ASTs, where if you're dealing with a multi-drug resistant gram-negative like these carbapenem-resistant Klebsiella pneumoniae in the study, sometimes it took up to 72 hours before our final antimicrobial susceptibility results were reported. Whereas with the nanopore real-time approach, we could have results within 31 hours, or the assembly approach within 36 hours. So the logical next question was, could we have an impact on patient care if the clinicians had those results to them within that 31 or 36 hours as we're estimating in the previous slide? So we did this hypothetical trial design to evaluate time to effective therapy. And so of the 28, there were 28 patients with carbapenem-resistant Klebsiella pneumoniae infections. 22 received inadequate empiric therapy, and the median time to effective therapy for conventional antimicrobial susceptibility results was 61 hours. Whereas if we would have applied a rapid whole genome sequencing approach to prediction, we had the potential to impact 20 of those patients, with a median time to effective therapy of 41 hours with the real-time approach, or 35 hours with the assembly approach. And you might be asking, well, why is the assembly approach quicker than the real-time approach? Well, when we said the turnaround time of the laboratory methods were longer with the assembly approach, the reason is that the assembly approach provided more comprehensive data to infer the AST activity than the real-time approach, for which there were delays on awaiting additional ASTs. So here we have two proof of concept studies showing that we can apply whole genome sequencing AST methods to potentially impact patient care. But how do we predict those phenotypes? Well, it's really not all that straightforward. And there are really two different approaches that have been applied in the literature. And at its essence, we're trying to predict susceptibility or resistance of an antimicrobial for a specific bug. The most common approach that has been applied are these rule-based approaches, where you use curated databases of AMR loci or SNPs, and then you try to find AMR genes or KMers from your data using those databases, using either raw reads or assembled genomes. But that's the first step. The first step is identifying the AMR gene. The second step is then to predict it, which is not always very straightforward. You need to correlate that gene or SNP to a phenotype, and it will vary by organism, expression, the interplay between multiple mechanisms, and by antimicrobial agents. The second approach is this model-based approach, which we're starting to see more publications on, which uses machine learning or statistical approaches, where you can link the entire genome or specific markers to an MIC or a susceptible resistant result for each antibiotic and train the models to try to predict those results, the MIC or the susceptible or resistant results. These models have the ability to predict MICs with no a priori information about the underlying gene content or resistant phenotypes of the strains. So the models really do that in the background to pick out the important information for those predictions. So the next step is, once you've predicted it, you're going to have to compare to the phenotypic reference standard, which is broth microdilution, because the intended use of whole genome sequencing is to predict your phenotypic AST, so you need to compare it to your reference standard. And many of these initial studies that I quoted throughout this presentation, especially that initial slide where I compared a bunch of them, they were using non-reference AST methods as their comparator. However, if we really want to get this methodology through the FDA, they're going to require the same standards as traditional phenotypic methods, where you're going to need to compare to a reference broth microdilution method, as well as get assess your results in terms of categorical agreement. Are you getting the correct susceptible or resistant categories? And then also further looking at the types of errors you're encountering. Are they major errors where you're detecting, you're calling something falsely resistant and you have to keep those below 3%, or very major errors, which are the errors that scare me the most, when you're calling something falsely susceptible when they're indeed resistant, and the FDA requirements there are less than 1.5% in terms of, so we're going to have to fit or have our predictions be within these similar expectations to what we have with phenotypic ASTs. The other problem is that the method that we're comparing to, the phenotypic reference standard, is an imperfect standard itself, where the standard dilution is plus or minus 1 doubling dilution. So I show at the bottom of the screen here, the MIC is red, where you see visible inhibition of growth here at 0.5. But if you repeated this in triplicate, you might, within the standard error AST, might get a result of 0.25, 5, or 1. And this can vary significantly more depending on the organism or antimicrobial agent combination. For instance, with piperacillin tazobactam and Pseudomonas aeruginosa, the variability can be much larger just within the standard error of AST testing. Furthermore, and so this variability is really not accounted for in the clinical setting, as it's not practical for us to perform replicate MIC tests for clinical care. And so these prescribed methods are highly standardized from inoculum density, media, temperature, atmosphere, and incubation period, the phenotypic methods, which really do not reflect the variation of the environment and expression of phenotypes that can occur during clinical infection. So really what we need to do is also look at doing some clinical outcome studies to look at the value of WGS AST predictions in terms of clinical outcomes in patients. And so hopefully we'll start to see some of those studies coming down the pipeline. So getting back to our prediction pipeline, our prediction methods, so the initial rule-based approaches that were applied by many of the studies apply many assumptions to the data. First is that there's a single genetic locus that's responsible for the phenotype, or if multiple loci are present, they do not interact. Second is that these loci are highly penetrant and are not affected by the strain background. And the third is that there's this complete knowledge of the genetic basis for that particular phenotype, which we know is not true. And we know that the mechanisms mediating resistance can be complex, just as I explained in that first case that we had, that there are heterogeneous mechanisms that can result in sabituril resistance, for example. And so what the model-based approaches attempt to do are these studies attempt to capture that uncertainty in the genetic basis of resistance and reduce overfitting of that data using a variety of statistical modeling and machine learning approaches, as I've highlighted in the red box here. The good thing about these model-based approaches is that the accuracy seems to be really well, and excitingly, we could see the ability to predict the minimum inhibitory concentration, or the MIC, within ±1 doubling dilution, which is amazing, and that's what we would expect within the standard error of phenotypic ASTs. So the next step that we need is, so we're addressing methods, we're addressing predictions of the phenotype. The next is that we need simple analysis tools, especially for introduction to clinical microbiology labs to help for patient care, because we really don't have the bioinformatics expertise, nor do we have the time to do some of these manual curations. And so what we're starting to see are these fully automated analytic programs that are suitable for use in clinical microbiology being introduced. I list three here: the Next Gen Diagnostics, 1928 Diagnostics, and Aris Genetics, where the first two have been applied and published in the literature on applying these rule-based K-mer analysis tools specifically to Staphylococcus species. Whereas some of the Aris Genetics studies and some of the studies that I've been able to collaborate with them with, which I'll go over in a few slides, we've started to look at a variety of organisms, and it uses the stack ensemble models and rule-based analysis using the Aris database. And so we'll go into that a little bit further. But here's one of the publications comparing next-gen diagnostics and the 1928 analytics programs for 102 Staph aureus isolates recovered from positive blood cultures to try to predict the phenotypic profile for these isolates, including these agents listed here. There are some differences in which one of the analytics could predict some, but not all of the agents, and hence the difference in the combination of isolate antibiotic combinations that can be compared between these two pipelines. But overall, it was really encouraging to see a high concordance between the predictions and the phenotypic AST results, where there was 100% agreement with oxacillin, vancomycin, and mupiricin for all three for these pipelines. But you do see that there were some discordant results that occurred, and that if you look at the error rates associated with those discordant results, they would be outside of what would be acceptable for a FDA-cleared assay. That being said, this study really showed that these automated bioinformatic tools may be useful for a clinically amenable workflow to predict the genotype, both phenotypic susceptibility and resistance in Staph aureus. Now these studies have been applying Illumina-based technologies, and what we wanted to do in our research lab is see, can these analytics handle nanopore-based data, because nanopore-based data can be produced, as we said, in real time, has the potential to be available to impact patient care. However, what I find most of the time is that some of these programs are developed specifically for Illumina data and cannot be directly applied to nanopore data. So what we did in collaboration with Aris Genetics is took 144 of our Klebsiella pneumoniae, including carbapenemase and non-carbapenemase producing, as well as carbapenem susceptible isolates. And as you recall, in my research lab, we do that hybrid assembly. So we have both Illumina and nanopore sequencing results for all of our isolates to help us with genome assembly to define resistance as well as mechanisms of spread, etc. But we use this data here to assess how the pipeline would do with Illumina data on its own and with nanopore data on its own. And so we use the Aris Genetics Cloud pipeline, compared to broth microdilution, and created 1,982 comparisons between the predicted and observed resistance phenotypes for 19 antimicrobials. And what was really exciting is that we found predictive AST demonstrated comparable results for both nanopore and Illumina data, which is exciting because, again, although the nanopore data has a higher error rate, it looks like some of our bioinformatics tools and analysis pipelines can help us overcome that and allow us to kind of use these tools for prediction for real time in the clinical setting, where an overall agreement that we've seen was 89% for Illumina for categorical agreement versus 88% for nanopore. So very comparable, but we also evidently saw better performance with our carbapenemase producing organisms at 95 and 96% categorical agreement. And it was slightly lower with our significantly lower for our non-carbapenemase producers, and that's because it could be more difficult to predict carbapenem resistance in the absence of that acquired resistance gene. But we have been working along with our Aris collaborators to further tweak the ability to define carbapenem resistance among those non-carbapenemase producers. The other thing that comes up is that we're now starting to see treatment guidelines based on whether mechanisms of resistance testing is performed or not. And these treatment guidelines recommend therapy. The recommended therapy will differ based off of the mechanism mediating resistance. So for example, if your phenotype is third generation cephalosporin resistance, you don't know based off of the phenotype whether that's mediated by ESBL or AMP-C. That being said, if you knew the mechanism mediating resistance, the recommended treatment would differ, where with ESBL, meropenem would be the recommended therapy versus AMP-C cefepime. So not only understanding the phenotype, but knowing the mechanism mediating that phenotype can be helpful. And the same with carbapenemase producers, where based off of whether it's a carbapenemase producer or not and the genotype, the recommended therapy will vary. So we've been working with Aris again to try to merge the predictive AST and that genotypic information to try to more accurately guide therapy. And we recently put this data together, looking at the combination of predictive AST and genotypic information among a group of Enterobacteriaceae, so expanding past the Klebsiella pneumoniae that we had focused on before and applying it to E. coli, other Klebsiella species, Enterobacter cloacae. And so here in this study, we were able to demonstrate the accuracy of the Aris Genetics pipeline for prediction using both Illumina and nanopore sequencing with a broader set of gram-negative bacilli, but also this overall accuracy of 93% versus 91%. By combining these two approaches, which will have the potential to further help guide therapeutic management of patients. The last study I'm going to highlight here is a study, again, that applied the Aris genetic database to a wide variety of gram-negatives as well as Staph aureus to predict AST. And you can see here that overall the categorical agreement varied by organism as well as antimicrobial agent, but showing that we can apply these pipelines more broadly to potentially be applicable for patient care. And I guess the last question that we have, so we're working through the methods, the predictions, the analysis, is the last thing is, well, how do we report these results appropriately to positively impact patient care? In general, these results that we've been approaching it as susceptible and resistant categories and grouping the intermediate with the resistant category. However, the question comes up, are we confident enough to predict susceptibility? You know, like I said earlier, the error that I'm most worried about is a very major error where we're telling a clinician that this isolate is susceptible when in fact it's resistant. So is there a different wording or a softening of the wording that we could potentially put in our reports prior to the phenotypic ASTs being available? And then there's also discussions about whether you could put accuracy estimates or reliability scores around these results to kind of guide the clinicians in terms of the expectation of how accurate those predictions are. So what do we need at this point to more broadly apply whole genome sequencing to predict AST in the clinical microbiology lab? I've kind of went over these a little bit already, but again, just to reiterate, more rapid turnaround times, simplified workflows with reduced hands-on time, this organism to sequencing robotics or microfluidics. It looks like next-gen diagnostics are working on some sort of microfluidics to apply for strain typing purposes for whole genome sequencing, but you could technically apply that same type of method for predicting AST. We need more cost-effective approaches, and this is being done through both Nanopore and Illumina. Nanopore has a flongle that's only $90 for the flow cell, so we're coming into a more relevant cost analysis compared to some of the broad molecular panels that we might implement in the lab. Or from the Illumina side, they have different sequencers now, like the iSeq and the MiniSeq, that can allow for more rapid sequencing times. And then we need tools that will convert that live streaming data, and we need working groups to discuss the best methods to convey those results, as well as outcome studies to determine the clinical accuracy and impact to patient care if we were to start to apply this clinically. So in summary, whole genome sequencing allows us to define mechanisms of resistance and to detect all antimicrobial resistance genes harbored by an isolate, or also known as the resistome. Whole genome sequencing predictions has been demonstrated as an accurate tool to define and predict resistance among a variety of organisms. Improvements around methods, turnaround time, prediction methods, and automated analytic tools are bringing us closer to implementing whole genome sequencing to predict AST in clinical labs. And these automated analytic tools are being adapted for use with both Illumina or Nanopore data, which is great, such as was shown with the research that I summarized here with Aris Genetics. And there's still some work ahead before these assays can be FDA cleared for clinical use, where there needs to be further consideration on reporting, and clinical outcome studies are required. So thank you so much for your attention and for listening to me today. If you have any questions, feel free to reach out to me. Otherwise, I'm happy to take any questions.
Okay. Thank you, Dr. Simner, for your informative presentation. We will now start the live Q&A portion of the webinar. If you have a question you'd like to ask, please do so now. Just click on the Ask a Question box located on the far left of your screen. We'll answer as many of your questions as we have time for. So let's get started. Our first question is two parts. Any idea why cephiderical resistance is mainly seen with NDM producers and not other MBL producers? And how exactly did you notice the multiple copies of NDM5?
Thank you for the questions, and thank you everyone for listening in. It's great to see the global audience asking some questions. So hi everyone. So to answer the questions, the first question is that I think some of our NDM producers have some hydrolysis rate, baseline hydrolysis rates of NDM. And maybe the reason why we're seeing some resistance among NDM producers, but more specifically, I think it might come down to the specific allele What we've seen in our lab is NDM5, like in the case I presented, tends to possess enhanced carbapenemase activity. And so I think not only is it just NDM, but also the allele might become important when thinking about cefidorachol resistance and NDM producers. And in terms of how did we determine the copy number, as I mentioned, we used both long read and short read sequencing methodologies, where the long reads allow us to actually kind of close the loop on those plasmids and sequence the entire plasmid. And we could see with sufficient coverage and multi-locus sequence alignment that we were indeed seeing increased copy numbers within those plasmids in the later isolates after the patient had been on cefidorachol therapy.
Thank you. Next question. What are your thoughts in terms of the possibility of discovering alternatives to novel drugs, for example, repurposing existing drugs by combining them in novel ways to preserve the last resort drugs?
It's a good question. So I think there's two things here, right? How can we use the data that we get from whole genome sequencing and understanding mechanisms of resistance to discover alternatives or new drugs? We can try to devise new agents that can overcome some of the resistance mechanisms that we're defining through whole genome sequencing and further defining through functional studies. The second question is, the second part of that is, there's certainly studies now looking at repurposing older agents. If, for example, phosphomycin, traditionally thought as a urinary drug, is now being approved for IV formulation, I think mostly outside of the US for now. So our European colleagues might be seeing that where, it's an older drug that they're trying to utilize now and may have activity against MDR gram negatives. Or in the example of our case, where the combination of two approved agents may be able to overcome the mechanisms mediating resistance, such as aztreonam combined with ceftazidime avibactam. And I know that the combination of aztreonam avibactam itself is undergoing further clinical trials currently. So again, looking at learning the mechanisms can help us understand how we can reuse, repurpose some agents that we might otherwise disregard just based off of the phenotype, where we can combine them or revisit their use for other purposes, or use that data or that information to try to create new agents to overcome those mechanisms.
Thank you. Next question. You showed a comparison of several automated analytics and pipelines. How are these technologies different, and what differentiates ARIS genetics?
Yeah, I think first off, I don't have too much experience with the other analytic pipelines other than ARIS genetics, as I highlighted in the slide there. And so most of my information in regard to the 1918 diagnostics and the next-gen diagnostics are what's published in the literature and what they have published on their websites. But the two things that kind of stand out on what's available currently in the literature about these methods is one is that, to my understanding to date, most of the publications on 1918 diagnostics and the next-gen diagnostics have only applied Illumina data, and the publications have focused mostly on Staph species, or even more specifically on Staph aureus. Whereas with the ARIS pipeline, and again with some of our research and collaboration with ARIS, is that we found that we can apply that pipeline with comparable accuracies with both Illumina and Nanopore data, but also we're starting to broaden application to a broader range of drug-bug combinations, so a broader range of organisms to not just be focused on a single pathogen. So that is what I'm aware of so far based off of the literature. So those would be the main differences I would highlight.
Thank you. Our next question has two parts. What are the pipelines that can be used to predict ARGs and its mechanisms from pathogens? And can ARGs be predicted from metagenomic sequences?
Yeah, I think the pipeline, so there are many different databases and programs out there that can just easily detect antimicrobial resistance genes, right? There's CARD, there's oh my gosh, it's slipping my, but there's many of them that are available out there. And what those pipelines do are simply detect the presence of a gene. I think what we really need is not only detection of resistance, but prediction of phenotype. And so that's where these automated analytics like the three I presented in this presentation are helpful, where it's not just detecting the resistance gene or associating resistance or the potential of resistance. These are actually being developed to predict the phenotype. And then in terms of metagenomic sequencing, it's a little bit more difficult, right? Because you have, depending on the source, you might have a whole lot of competition in the background. You might not be able to know who the gene belongs to. So I still think we're a little ways out on predicting, phenotype based off of resistance genes from metagenomics. We can certainly detect them, but being able to predict phenotype is a whole other can of worms that are still being developed.
Thank you. Next question, can you please comment on how is the RS technology already used today in clinical routine?
Yeah, I think so. In my opinion, it's really still in the research use only setting, from what I can tell. I'm not sure if there's any other. I haven't found any publications of them really applying it clinically currently. That being said, ideally we could start to get those studies out there that that applies this in the clinic, in the clinical lab, so that we can not just hypothetically show its value, but show its value moving forward, like combining nanopore with an automated analytic like Aris Genetics.
Thank you. Next question. Was the cost analysis of WGS per colony comparing with other methods performed?
So we didn't do a cost analysis currently in our studies. However, I'm optimistic that these costs are coming down. If we think about nanopore and the flongle, it's $90 for that flow cell, and we can start to get the reagents for a whole genome sequencing sequence down, comparable to some of the multiplex molecular panels that we apply. So I do think that the costs are coming down, and whether you're able to multiplex these would further help you reduce those costs. And similarly with Illumina platforms, we're starting to see these smaller sequencing platforms that allow for more real-time or like shorter sequencing runs, but as well as reduce costs for the flow cells, etc.
Thank you. We have time for one more question. What do you see as the next steps? Which disease states, sample types, pathogens would you see the most utility in for predictive AST?
That's a great question, and one of the studies that we're currently working on is looking at blood culture isolates. Right now, we're starting to see the introduction of kind of liquid inoculums, whether that be through a sepsityper or other methods that are becoming available. So with blood culture, the advantage there is that most of the time they're monomicrobial, and if you can get sufficient biomass from that positive blood culture, that would allow us to even further reduce that turnaround time in terms of predictive AST. And so that's one thing that we're planning to work on a little bit further as our next study. And to kind of tie together some of the other questions, we're going to work on using the flongle, try doing this more in real time along with clinical care to assess how well are we doing if we thought about this in terms of workflow in the lab, as well as how well are we doing in terms of cost comparison from FTE standpoint and reagent standpoint as well.
Great.
So that's where we're going with that. No worries.
Well, thank you again, Dr. Simner, for your time today and your important research. We would also like to thank LabRoots and our sponsor, OpGen, for underwriting today's educational webcast. Before we go, I'd like to thank the audience for joining us today and for their interesting questions. Questions we did not have time for today, and those submitted during the on-demand period, will be addressed by the speaker via the contact information you provided at the time of registration. This webcast can be viewed on demand. LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Until next time, goodbye.